So in this video, we are going to continue talking about microbial metabolism, and specifically we're going to start to dive in and talk about the types of metabolism. So we'll compare aerobic respiration, anaerobic respiration, and fermentation and go into a little more details. Now, when you're studying this for my class, what you need to be aware is that I don't care so much that you get bogged down with the little details, meaning glycolysis has 10 enzymes, 10 steps. I'm not going to ask you that. I'm not going to ask you what are each of the intermediates along the way. I'm not going to ask you, what are all the enzymes. What you want to focus on when you're studying for this part is for each reaction, so like for glycolysis, what goes in, meaning what are the reactants, what comes out, what are the products, what are those products used for, meaning do they go to the next step in the process, are they waste products, basically what are those products used for, and then where does it take place. And so we're going to walk through and kind of do an overview. And hopefully my goal with this with you guys is not to get you to memorize details, but to understand the big picture. What is the purpose of doing all these steps? Why do cells go through this series of steps? So in order to understand cellular respiration, we have to understand redox reactions. And you guys started to learn about redox reactions in lab because we did a lot of tests that looked at redox reactions. We talked about our anaerobic jar, right? And so we had to reduce O2 to water to make the jar anaerobic. We did nitrate reduction. So we've already talked a little bit about redox reactions, but we're going to review it here. So a redox reaction is an oxidation reaction paired with a reduction. And so what that means is that electrons are basically redistributed. One molecule or ion is going to contain the electrons, and it's going to pass those electrons to something else. So in lab, you might recall that I taught you guys oil rig. And that is that an oxidation is a loss of electrons, and a reduction is a gain of electrons. Oops, we don't need that one there. Reduction is a gain. So oxidation, again, is going to be the molecular atom that is going to lose the electron. So notice in this bottom diagram, we have X. Substance X has the electron. So when it gives its electron to Y, X becomes oxidized and Y becomes reduced, meaning it's going to gain the electron. So all we're simply doing in this case is we're redistributing the electron. We're transferring it from one substance to another. And so that's why you always have oxidation and reductions being paired, because the electron has to come from somewhere. One atom is going to donate, one is going to receive. And so that's going to be our redox reaction. So, question for you to see if you understand your redox reactions. When sodium, atomic number of 11, and chloride, atomic number of 17, come together to form sodium chloride, NaCl, what has happened to the sodium? Is it red? It became oxidized. Yellow, it became reduced. Or green, can't tell from the information. So my hint to you when trying to solve this, draw out the electron shells. See how the electrons are going to move, and then that's going to help you with whether sodium becomes oxidized or reduced. So when you get a second, pause your video, and when you're ready, once you've tried to work through it yourself, go ahead and restart the video to get the answer. All right, so let's go through this. So we have sodium. Sodium has an atomic number of 11. Remember, if its atomic number is 11, how many protons does sodium have? And the answer is 11, because the atomic number is equal to the number of protons. So sodium has 11 protons. Protons carry a positive charge. So if sodium has 11 protons, how many electrons will sodium have total? And the answer is 11, because the positive charges and the negative charges balance each other out. So if we're looking at our electron shells, how many does sodium have in its first shell? 2, right? That's going to fill its first shell. How many in the second shell? 8. That will bring me to 10 total. And so how many in the third shell? And the answer is 1. So we have 2, 8, and then 1. That's my 11 electrons. So now let's do the same thing for chlorine. So chlorine has an atomic number of 17. How many protons? 17. How many electrons? 17. So how many in the first shell? 2. That's going to fill the first shell. How many in the second shell? 8. That brings me to 10. So how many in the third shell? So if we're at 10, that means there's 7 in the third shell. So 1, 2, 3, 4, 5, 6, and 7. So if you're looking at your electron shells between sodium and chlorine, how many more does chlorine need to fill its outer shell? And the answer is that chlorine needs 1 more. Sodium, on the other hand, right? Sodium has 1 valence electron. How many more would sodium need to acquire to fill its outer shell? And the answer is 7. That's not very likely. So sodium is not going to try and gain electrons. Instead, sodium is going to donate the electron. And when it donates, sodium becomes oxidized. It loses the electron. And chlorine becomes reduced because it gains the electron. So the answer is red. Sodium becomes oxidized. So when we look at cellular respiration is essentially one big redox reaction. It's simply relocating electrons from one molecule to another. And when we do this, we are going to extract energy, and we're going to talk about how. So if we look at our reactants, so what goes in for cellular respiration? We have glucose, C6H12O6, and we have oxygen, O2. And so those are my 2 reactants. That's what needs to go in for cellular respiration. Now, in cellular respiration, what we are essentially doing is we're taking our glucose and breaking it down stepwise. So we're going to start by taking that 6-carbon glucose and chop it into 2 3-carbon pyruvates. And we will just continue to break it down until we're left with a product, which is going to be our carbon dioxide. So we're going to break glucose down completely into the inorganic carbon dioxide. So what happens is when we break those bonds, the electrons are going to be released. And you're going to see in a minute that those electrons get put on an electron carrier. So we say that glucose becomes oxidized. It's going to lose electrons and lose hydrogens. Remember in lab that I told you that one of the things you can do to follow whether it's an oxidation or reduction is to look at hydrogens. So if hydrogens are removed, that means that electrons are removed. Because a hydrogen is going to be an H plus and an electron, and they're going to move together. So the hydrogens on the glucose are going to be transferred, and at the end, what's going to take those electrons and take those hydrogens is going to be oxygen, O2. So we say that O2 becomes reduced because it gains hydrogens. It's going to form water. So when oxygen acts as the final electron acceptor, O2 gets reduced. It's going to gain the electrons and it's going to form water. So notice that O2 gained hydrogens, which means that it also gained electrons. And by doing this process, energy is produced because remember, we've talked about endergonic and exergonic reactions. Breaking down glucose is going to be an exergonic process. Energy is going to be released because the energy is stored in those bonds. Food has what we call potential energy. It has the potential to do work. So there's energy stored in those bonds, and when we break those bonds, the energy gets released. And the energy that gets released is used to produce ATP. So now, thinking along these lines, right? Thinking about following hydrogens. If I was to ask you this, and let's say the reaction goes this way, meaning that this time carbon dioxide and water are the reactants. That's what's going in. And the products are going to be glucose and oxygen. What is the name of that process where we take carbon dioxide and water and we build glucose? And the answer is the name of that process is photosynthesis. So photosynthesis is the reverse of cellular respiration. So now, if I were to ask this in terms of photosynthesis, we have two reactants, carbon dioxide and we have water. Which of those two reactants is going to become oxidized? And which of those two reactants is going to be reduced? So again, if you need time to think, pause. Think about which one is oxidized and which one is reduced. So if we're talking about oxidized, right, that's going to be the one that loses the electrons, which means that it also loses hydrogens. So if you're looking at carbon dioxide and you're looking at water, right, which one has hydrogens? And the answer is the water. So in the reverse, the water, right, would become oxidized. And that does happen in photosynthesis. Water donates electrons to an electron transport chain. So water becomes oxidized, and the CO2, the carbon dioxide, is going to be reduced. It's going to gain hydrogens, gain electrons, and so CO2 becomes reduced. It's going to be reduced and it's going to build a sugar. Now, energy has to be put in for the reverse reaction to happen, right? So to build sugar, energy has to be put in. Where does that energy come from in photosynthesis? The answer, light, sunlight, right? Photo refers to light. So light is the energy that's driving that process. But notice that photosynthesis is also a big redox reaction. Something is donating electrons, something else is receiving it. So what we need to talk about next is for our redox reactions. In our redox reactions, we have coenzymes, and these coenzymes act as electron carriers. And so you might recall that a coenzyme is going to be organic in nature, and they are going to carry the electrons. Now, when the electron carrier is reduced, meaning it has the hydrogen, it is energy rich because those electrons are energy rich. So if a molecule has its hydrogen, it's energy rich. If it is oxidized, right, if it loses hydrogen, it's energy poor because those electrons have energy. And so if it does not have those electrons, it's energy poor. So there are several electron carriers that we're going to talk about. So the first one in its reduced form is going to be NADH. Notice it has its hydrogens. In the oxidized form, it's NAD+. So that's when it does not have its hydrogen, it does not have its electron. Another one that we're going to talk a lot more about as well is going to be our FADH2. And the oxidized form is FAD. NADH and FADH2 are both used in cellular respiration. And then we have NADPH, and the oxidized form is going to be NADP plus when it's oxidized, when it lost its electron. So the reduced forms are the ones that are energy rich. And the reason for that is that you're going to see in a little bit that when those electron carriers take those electrons to the electron transport, they're going to drop off electrons at the electron transport chain. That is where the bulk of the ATP comes from. So that's why these reduced carriers, these electron carriers that have their electrons, they're considered energy rich, because when they drop off those electrons are going to be used to produce ATP. So this is just showing you an example of a redox reaction. So the yellow blob is an organic molecule. So let's say glucose, for example, right? So glucose, remember, in respiration is going to be our, the glucose is going to become oxidized. It's going to lose its electrons. And so the way that that works is this organic molecule has these hydrogens. So notice that each hydrogen is written as one proton. So that's the black dot. And one electron, right? And so that's my hydrogen atom. And the electrons are going to be passed from the organic molecule and put onto the empty electron carrier. So again, I have a diagram in a minute. Empty does not have the electron. Right? So in this case, our empty electron carrier is our NAD+. So our organic molecule is going to donate electrons to NAD+. It's going to transfer its electrons onto NAD+. So our organic molecule then, our food, becomes oxidized. It loses its electrons. So loss of electrons. The NAD plus becomes reduced. This is a reduction because we've gained electrons. And so the way that that works is that this whole hydrogen, so one whole hydrogen gets transferred. So that is a proton plus the electron gets transferred. That's the NADH. So one whole hydrogen gets moved. And for the other hydrogen, only the electron gets transferred on the NAD+. And when only the electron is transferred, that neutralizes that positive charge. So notice that it's not NADH+, it's just NADH, because that other electron is going to neutralize that charge. And so the electron is going to go on to NADH, and the hydrogen ion, the proton, is going to be released. And you're going to see the importance of hydrogen ions later on. So when we talk about biological oxidations, so talking about our redox reactions where the organic molecule loses electrons, we often call those dehydrogenations, right? They become dehydrogenated. And that's because they are losing their hydrogen. And so the enzymes often that do this are called dehydrogenase. They're responsible for removing electrons from that organic molecule. So now we're going to look at the way that ATP is generated. And there are three basic mechanisms of phosphorylation that are used to make ATP from ADP. And so remember that ADP is adenosine diphosphate. So notice that it has two phosphate groups. And when we go to form ATP, we need to add a phosphate group onto ADP, forming ATP, which is adenosine triphosphate. So notice we have 1, 2, 3 phosphate groups. And so we're going to build ATP. So we call this a phosphorylation because it's the addition of a phosphate group. So we add a phosphate group to ADP, and when we add that phosphate group, what we get out is our ATP. Now, there's three main ways that ATP is produced. The first one is what we call substrate level phosphorylation. And this is simply transferring a phosphate from a substrate onto ADP, and that will form our ATP. So it's just like it sounds, substrate level phosphorylation. We're simply just going to take some substrate that has a phosphate group on it, and an enzyme is going to catalyze the movement of the phosphate from the substrate onto ADP, forming ATP, and that's a P. Let me make that a little more clear. The next type of ATP production is what is called oxidative phosphorylation. And oxidative phosphorylation uses redox reactions to form ATP. So an example of this would be the electron transport chain. That is going to couple redox reactions to the formation of ATP. And so that's oxidative phosphorylation. The last one, photophosphorylation. What does photo usually refer to? Light. So using light energy to form ATP. And what process would you guess uses photophosphorylation? That's going to be photosynthesis. And so we're going to look in a little more detail for each of these three methods of ATP production. So the first is going to be our substrate level phosphorylation. And just like we wrote on the last slide, all this simply is the catalyst of moving the phosphate from the substrate. So notice here's my substrate, and it has a phosphate group. And this specific enzyme is going to bind to the substrate and also bind to ADP. And it's simply going to transfer the phosphate from the substrate onto ADP, and what we get out is ATP. This is used in glycolysis and the Krebs cycle, meaning in those two stages, that is how ATP is produced. It's produced by substrate level phosphorylation. And so again, all we're going to do is transfer the phosphate from some substrate onto ADP, and what we get out is ATP. So that's substrate level phosphorylation. Next, we have our oxidative phosphorylation. And again, this is going to be a series of redox reactions using electron carriers. And it's going to release energy from these redox reactions that is used to form ATP during the electron transport chain. And so the electron transport chain is where the cell is going to utilize oxidative phosphorylation. And so this is going to actually generate the majority of ATP. This is going to be the highest yield. So you're going to see in a minute that when the cell does glycolysis, when the cell does the Krebs cycle, in each of those steps, we get 2 ATP per glucose. So the energy yield is not very high using substrate level phosphorylation. That is in contrast to the electron transport chain. And in the electron transport chain, right, we're going to get 26 to 28 ATP per molecule of glucose. So we get the greatest ATP yield in the electron transport chain. And then lastly, we have our photophosphorylation. Again, that's going to convert light energy into ATP and our electron carrier, which is NADPH. And then those two products during what we call the light reaction, those two products are then going to go on to what's called the Calvin cycle in photosynthesis. And that's where we're going to be able to build, the plant's going to be able to build its sugar. So again, used in photosynthesis. So photophosphorylation is used to make ATP in photosynthesis. So what we're going to do to start out is we are going to compare and contrast the different modes of metabolism. So what I'm going to start by focusing on is what is the difference between aerobic respiration, anaerobic respiration, and fermentation? What makes these processes different? So this slide is just summarizing and comparing the 3 different types of metabolism. Now, fermentation is going to give us the lowest ATP yield. It's going to give us 2 ATPs per glucose. This diagram here shows 36 to 38 with aerobic respiration. I'm going to cross that out. More recent studies, which actually has been done for a while, but textbooks have not updated this. More recent studies suggest that it's actually 30 to 32 ATP. So not the amount that's written in this diagram, not 36 to 38, it's actually 30 to 32. And so as a result, I'm also going to change the number for the anaerobic. So for anaerobic respiration, we get more than fermentation. So I'm going to put the low number being 3. Whoops. So 3, but not as much as aerobic respiration. So we're going to put 29. So we're going to change these numbers a little bit. Now, in anaerobic respiration, there's a lot of variability in terms of ATP yield. Notice 3 to 29, that's a pretty big distinction. Now, the other thing that I really want to point out is the difference between anaerobic respiration and fermentation. And there is a very key distinction. And in fact, when I looked online, almost probably 90% of the websites that were out there were using the term anaerobic respiration wrong. They were calling fermentation anaerobic respiration. That is absolutely wrong. It's wrong. Bacteria, for example, do true anaerobic respiration. Not many other organisms do. So if you don't study bacteria, a lot of people are using that term incorrectly. So let me compare what is different and what is the same between these processes. So for aerobic respiration and anaerobic respiration, whenever we call it a respiration, respiration uses an electron transport chain. So it uses an electron transport chain. So that's what these two have in common. They both have glycolysis, they both have Krebs cycle, and they both use the electron transport chain. The key distinction between aerobic respiration and anaerobic comes into the final electron acceptor. In aerobic respiration, O2 is going to be the final electron acceptor. On the flip side, we can use sulfate, nitrate, carbonate. Those are all inorganic final electron acceptors that are not O2. So inorganic final electron acceptor that is not O2. So notice that none of those molecules has both carbon and hydrogen. In both of the respirations, glucose is broken down completely to form CO2, which is going to be inorganic. So inorganic products. We are going to break down glucose completely. So we start with our 6-carbon sugar. It's broken down into CO2. Now let's compare anaerobic respiration and fermentation. So key differences between anaerobic respiration and fermentation. Fermentation does not use an electron transport chain, right? So that's a difference between them. A similarity is both are anaerobic. I said that wrong. Both are anaerobic processes. So without O2. So what they have in common is that they're both anaerobic, right? So that's what they have in common. They are both anaerobic processes, but apart from that, they're much more different than they are alike. So what I mean by that is that in this case, notice for fermentation, uses organic compounds as the electron acceptor. So instead of using an inorganic molecule, we're actually going to use an organic molecule. So our final electron acceptor is either pyruvate, if we're talking about lactic acid fermentation, or acetaldehyde, if we're talking about alcoholic fermentation. Both of these final electron acceptors are organic. And we end up with organic products. So we do not break down glucose all the way. We're not going to end with CO2. We actually end with organic products that still have a lot of energy left. So alcohol, so ethanol, lactic acid, etc. So notice that anaerobic respiration and fermentation are very, very different processes. They are both anaerobic, meaning they both occur in the absence of oxygen. But anaerobic respiration, remember when you hear respiration, think electron transport chain. Anaerobic respiration uses an electron transport chain. It uses an inorganic final electron acceptor that is not O2. And anaerobic respiration breaks down completely to CO2. Fermentation, right? Fermentation does not use an electron transport chain. It only does glycolysis and then goes directly to fermentation. It uses organic final electron acceptors, and it produces organic products. So you can clearly see that fermentation and anaerobic respiration are not the same. You have to be really careful in how you use this. If you call it respiration, again, think electron transport chain. It's going to have an electron transport chain. And so I just wanted to kind of compare and contrast these three different modes of metabolism. And we're going to break it down and we're going to go through each one in more detail. So again, this slide is just comparing different types of respiration. And so when we're looking at cellular respiration, again, glucose is completely broken down. And when glucose is broken down completely, it results in inorganic end products, meaning we're going to get carbon dioxide and water. Now, respiration may be aerobic or anaerobic. In both cases, it uses inorganic final electron acceptors. If we call it aerobic respiration, oxygen is the final electron acceptor. And this again is going to produce the largest amount of ATP. This is going to give us the highest ATP yield, about 30 to 32 ATPs per glucose. In the case of anaerobic respiration, we use an inorganic molecule other than oxygen to serve as our final electron acceptor. And so there are three main types of anaerobic respiration. So nitrate, you might recall when we did our lab and we did our nitrate reduction. What does nitrate get reduced to? Remember, if it's reduced, it means that it gains electrons. So what does it get reduced to? So if nitrate is the final electron acceptor, it's going to produce nitrite. And so our nitrate reduction test was seeing if nitrate could act as the final electron acceptor during anaerobic respiration. So we have nitrate can be our final electron acceptor. We can have carbonate, and carbonate will be reduced to form methane, CH4. Sulfate gets reduced to form hydrogen sulfide, H2S. So hydrogen sulfide. And for hydrogen sulfide, what test did we do that was basically looking at this? What test did we do that we were seeing if hydrogen sulfide is produced, meaning if sulfur was reduced? So that would be our TSI, right? Our TSI was looking at if sulfur can be reduced to form the hydrogen sulfide. And so what we were essentially testing was, can sulfate be the final electron acceptor during anaerobic respiration? This results in less ATP than aerobic respiration. So again, we're going to say between 3 and 29 ATP per glucose. More than fermentation, but less than respiration, than aerobic respiration. So next we have fermentation. And again, in fermentation, glucose is incompletely broken down. It is not broken down completely. It results in organic end products, right? We still end up with organic products. And so our organic products can either be alcohol or lactic acid. Now, this is an anaerobic process, meaning it occurs in the absence of oxygen. But again, it is not anaerobic respiration. This results in the fewest number of ATP. We only get 2 ATP per glucose. So it is the lowest energy yield. And because those products are organic, they still have a lot of energy left. So they're not done. Because they get the fewest ATP, these are organisms that are going to be slow growing. Their generation time is a lot longer because they don't get enough ATP to reproduce rapidly. So in this next part, we're going to focus on aerobic respiration. So in eukaryotic cells have cellular respiration take place in the mitochondria. And so if you recall when we talked about mitochondria is a double membrane system. It has this smooth outer membrane, and it has this extensively infolded inner membrane, which is also referred to as the cristae. And so the cristae is just another name for the inner membrane. Now, the reason that the cristae has all of these infoldings is because structure and function are related, meaning that there's a reason that that inner membrane is extensively infolded. And that's because the last step of respiration is the electron transport chain, and that happens in the cristae. So there are these proteins embedded in the membrane, and that's where the electron transport chain is going to occur. So if we have all these infoldings, we increase the surface area of that membrane, which means we get a higher ATP yield. So there is a reason that the inner membrane has these inner foldings. Now, in terms of compartments, because we have this two membrane system, the matrix is the space inside the inner membrane. And we have the space between the two membranes, which is called the intermembrane space. So we have two compartments, and that's going to come into play when we start to talk about the electron transport chain. We're going to talk about where each of these steps happens. Now, interestingly about mitochondria, remember that mitochondria are believed to have once been free-living prokaryotic cells, right? And we talked about all the reasons why, right? Mitochondria have circular DNA. They have some of their own DNA. It's circular like prokaryotic cells. The sequences are similar to prokaryotic cells. They have their own ribosomes, meaning they can make some of their own proteins. Those ribosomes are 70S ribosomes, just like prokaryotic ribosomes. The ribosomes in your cell are 80S. And the list goes on. And so it's believed that the mitochondria was once a free-living prokaryotic cell. Now, in terms of how cellular respiration plays out, in eukaryotic cells, cellular respiration begins in the cytoplasm and finishes in the mitochondria. So glycolysis is going to happen in the cytoplasm, but the Krebs cycle and the electron transport chain all are going to take place within the mitochondria. Now, do prokaryotic cells like bacteria have mitochondria? And the answer is no. They don't have membrane-bound organelles. So in prokaryotic cells, all of the early steps, so glycolysis, the Krebs cycle, are going to happen in the cytoplasm, and then the electron transport chain needs to happen across a membrane. In prokaryotic cells, what membrane will they use? And again, we talked about this. They will use their cell membrane because that is their membrane that they can use. This slide is just summarizing cellular respiration. And so glucose, C6H12O6, glucose is a 6-carbon sugar. It is polar, right? Think of table sugar, which is sucrose. If I put sugar into coffee, will it dissolve? The answer is yes, because sugar is polar, right? It's able to interact with water, which is why it's able to dissolve. So what that means is that glucose can't cross the cell membrane on its own. It can't get in on its own because it's polar. It can't get through the hydrophobic middle of the cell membrane. So in order for glucose to get in, Glucose has to be transported using a glucose transporter. So it's going to transport the glucose in. Now again, what happens is that respiration begins in the cytoplasm. And so what we're going to do is we're going to take the 6-carbon sugar. Each of these dots represents a carbon. And in glycolysis, glyco, sugar, lysis breaking, we are going to chop that glucose in half. And we get 2 3-carbon pyruvates. Now, during glycolysis, we do get 2 ATP, and we're going to get that by substrate level phosphorylation, which you'll see in a minute. One of the things you're also going to see is for each of these steps, when we're breaking bonds, we are going to get electron carriers. So in addition, one of the other things that is produced during glycolysis is NADH. Remember that that is my reduced electron carrier. It has its hydrogen, it has its electron. So that is going to be a very important product that's produced during glycolysis. Pyruvate is going to go through a transition step as it's transported into the mitochondria. And when it does this, it's going to break down more. CO2 is released at this point. And the remaining 2-carbon fragment has a coenzyme A added to it. And those will form 2 acetyl-CoAs during this transition. The acetyl-CoAs will then go into the citric acid cycle, which is also known as the Krebs cycle. Those 2 names are used interchangeably. And what we're going to do is to the remaining 2-carbon fragment, we are going to break it down completely. And so during this process, we're going to get 4 CO2s, 4 carbon dioxides. That's the remaining 4 carbons. We get 2 ATP by substrate level phosphorylation. And because we broke down a bunch more bonds, we are also going to get our electron carriers. And in this step, we get NADH and we get FADH too. Now, the first 2 steps, the glycolysis and the Krebs cycle, notice only give 2 ATP per glucose. The electron transport chain, so oxidative phosphorylation, is going to give us between 26 to 28 ATP. So the highest yield is going to come from the electron transport chain. And so what that tells us is that those early steps are not as important for the ATP that's produced because they produce very little ATP. But actually the important products that get produced during these early steps are going to be those electron carriers, because those electron carriers, when they go to the electron transport chain and they drop off electrons, those electrons are going to be used to produce the bulk of the ATP. And so what we put in for cellular respiration is going to be our sugar. We also need our O2 because O2 is going to be our final electron acceptor. What we get out is carbon dioxide. So when we break down the sugar completely, each of those carbons is released as a CO2. We're going to get water, and the water comes from when oxygen acts as the final electron acceptor. And also the E is going to be energy because we get ATP. So we need to talk a little more about electron carriers. And so again, you can think of electron carriers as electron taxi cabs or electron Ubers in these days. And so when the cab is empty, so the empty state of our electron carriers would be like for the NAD+, right? So NAD+ is empty. It doesn't have its passengers yet, and its passengers being the electrons. So NAD+ is empty, and it needs to pick up its passengers. And so its passengers are going to be electrons. Where do those electrons come from? Well, they come from the food that we eat. So when we break down the glucose, remember that glucose becomes oxidized. And so glucose and the molecules, the intermediates after glucose, are going to donate hydrogens and electrons and put them onto NAD+. And so again, like we saw in the dehydrogenation slide, the way that this works is one whole hydrogen is transferred. So that would be, let me put on my laser pointer. So this whole hydrogen gets transferred onto NAD+. And remember that the second hydrogen, the electron gets transferred and the proton is going to be released. And so now NAD+ became reduced. It gained electrons, it gained hydrogens, and again, those hydrogens and those electrons came from the food, right? And so the food becomes oxidized. So notice that it became oxidized, and the NAD+ became reduced. It gained the electrons. Now, once NAD+ has its passengers, meaning it's loaded, it has its NADH. Once it has its passengers, now those passengers, or the electron carriers rather, the NADH is going to take the electrons and it's going to drop off electrons at the electron transport chain. And so you can see here, so these electrons get dropped off. These hydrogen ions get released. You'll see what those do later on. And then we get back to being NAD+. So now the taxi cab is empty. It's ready to go back and pick up a new set of passengers. And so in the cell is constantly cycling between NAD+ and NADH. And again, it's when it has its electrons, when it's in the NADH form, that this molecule is energy rich, because when they drop off electrons at the electron transport chain, those electrons are used to generate the bulk of the ATP. And so this is kind of an overview of how our electron carriers work. So again, when you are studying cellular respiration, you want to study for each step what goes in, what comes out, and where does it take place. You do not need to know all of the intermediates along the pathway, okay? You just need to know what goes in and what goes out. So what goes in? Our input, we need to put in a glucose, and glucose is a 6-carbon sugar. So we put in our glucose, and even though this reaction is exergonic when we break down glucose, we still have to invest a little bit of energy to get the reaction going. And so 2 ATPs have to be put in, and they get hydrolyzed to form ADP, and that will get the reaction started. Now we're going to break the bond in the glucose, and we're going to break it into 2 3-carbon fragments. Now electrons during this process are going to get transferred onto NAD+. So remember I said that the food, so the sugar, is going to become oxidized, and it's going to put those electrons onto NAD+, and we get the reduced form, which is the NADH. And so that's true for both of these 3-carbon sugars. Now, the other thing that's going to happen is that you're going to get 2 ATP for each of these 3-carbon sugars. So notice that your products are going to be 4 ATP, but you had to put in 2 ATP to get the reaction started. And so we say that you get 2 net ATP. So you gain 2 ATP during this process. And again, that's because it requires energy to get the process started. And so the net gain is 2 ATP. These ATP, remember, are produced by substrate level phosphorylation. So notice that this substrate has these phosphate groups on it, and those phosphate groups are going to be transferred onto ADP, and we're going to form ATP. And so our other products that we're going to get that are important are 2 pyruvic acids or pyruvates. Those names are kind of used interchangeably. So we end up with 2 3-carbon sugars, and then the pyruvate is going to go into the citric acid cycle, meaning that the sugar is going to continue to be broken down little by little. In terms of the water, don't worry about that as much. But the products that I want you to be aware about, so pyruvate, you get 2 pyruvates, you get 2 ATP, and you get 2 NADH. And again, these NADH are going to be used in the electron transport chain. So what I did is normally in class, I would draw this out. but I've already drawn part of it for you, and I'm going to walk you through this. This diagram, as I go through the aerobic respiration talk, I would study this diagram. And if it's me and I'm trying to study for the exam, I would try and recreate this diagram from memory. If you can do that, anything I ask you related to this topic is going to be easy. You're going to have no problem whatsoever. So use this as a tool. This is going to be a summary of all the steps of cellular respiration, and I've color-coded the different parts to try and make it a little easier on you. So here we have our glucose. And glucose, remember, is a 6-carbon sugar. And so again, the first step in cellular respiration is referred to as glycolysis. Glyco, sugar, lysis, breaking. And so what that means, again, is that we're going to break the sugar in half. Now, where does it take place? So notice I've color-coded prokaryotic cells, that's the P. Prokaryotic cells, this happens in the cytosol or the cytoplasm. Eukaryotic cells, this also happens in the cytosol. So in both cases, this process is going to begin in the cytosol, happens in the cytoplasm of the cell. So in glycolysis, again, we're going to break that sugar in half, and what we get out are the 2 3-carbon pyruvates. We also, when we break bonds, you're going to see this every time we go through this and we break bonds, electrons are going to be transferred onto electron carriers. So we put in 2 NAD+, and when the electrons get put on my product, I'm going to get 2 NADH. Also, during this process, 2 ADPs need to be put in, adenosine diphosphates, and 2 ATPs are produced by substrate level phosphorylation. So when you're looking at this diagram, everything on this side of the arrow are things that go in. Everything by the arrowhead are things that come out, meaning those are the products. So these are what you need to go in, and these are your products. So for each of these steps, you need to study the name of the step, where do they take place, what goes in, here's our what goes in over here, and what comes out. These are my products. So keep that in mind when you're studying this. This is what you want to use as your tool to help you understand respiration. Now, remember that I said that in eukaryotic cells, for example, in eukaryotic cells, respiration begins in the cytoplasm or the cytosol, and it ends in the mitochondria. So what that means is that the pyruvate that we produce during glycolysis needs to get transported into the mitochondria. Now, notice that this is charged, right? There's this charged region. So pyruvate doesn't cross the membrane on its own. It needs a transport protein to help transport it. Now, when this happens, when pyruvate goes into the mitochondria, we are going to break off a carbon and we get CO2. So per pyruvate, we get 1 CO2. To the remaining 2 carbon fragment, so these 2 carbons here, to the remaining 2 carbon fragments, coenzyme A is going to come in and it's going to attach to that remaining 2 carbons. So here's those remaining 2 carbons from pyruvate, and they get a CoA added to them. And so now those products are acetyl-CoA. So we get an acetyl-CoA per pyruvate. Notice breaking bonds, we're also going to get our electron carriers. So NAD+ is going to pick up electrons and it's going to form NADH. So our products for each pyruvate, we get 1 CO2, we get 1 NADH, and we get 1 acetyl-CoA. So per glucose, how many CO2s do we get? And the answer is 2, because per glucose, so 2 pyruvates per glucose So we get 2 CO2s per glucose. We get 2 NADH per glucose. And we get 2 acetyl-CoAs per glucose. So notice this part's easy. Everything is multiples of 2. And so this step is not technically a separate step. This step has several different names for it. Sometimes you'll hear it referred to as the transition step. Sometimes you'll hear it referred to as the intermediate step. Sometimes you'll hear it referred to as pyruvate oxidation. There are a lot of names for this process. And so this is basically as the pyruvate is going into the mitochondria, but before the Krebs cycle. So this is like a transition between the 2 steps. So if we look at our diagram, we're adding on. So here are the pyruvates that formed from glycolysis. And so what's going to happen now is we are going to break those bonds. That's where the 2 CO2s came from, right? So this is going to be a CO2, and this is going to be a CO2. So there's my 2 CO2s that get produced. Broke some bonds, also get some electron carriers. So NAD plus goes in, 2 NAD plus goes in per glucose, 2 NADH come out. So now we have electrons transferred onto our electron carriers. To the remaining 2 carbon fragment, right? So to the rest of the sugar from pyruvate, the 2 carbon fragment, we get CoA, coenzyme A added on. So our product is going to be 2 acetyl-CoAs. Each acetyl-CoA is 2 carbons. So notice we still have 4 carbons left. Now I numbered this 1B. It's not a separate step, so I'm not going to call it step 2. It's the transition or the intermediate between glycolysis and the Krebs cycle. In prokaryotic cells, this is going to happen in the cytosol. In eukaryotic cells, this is as the pyruvate is going into the mitochondria. So pyruvate is going to go into the mitochondria. That doesn't happen in prokaryotic cells because remember that prokaryotic cells do not have membrane-bound organelles. So that going into the mitochondria is distinct for eukaryotic cells. So what goes in? 2 pyruvates, 2 NAD+. What comes out? 2 carbon dioxides, 2 NADH, and 2 acetyl-CoAs. And so again, when you're studying this or when you're taking notes on this, I would actually take the time yourself and draw it out. The act of writing it out is going to help you remember it. So this is going to be our transition step. Now, step 2 is going to be the citric acid cycle. Now, when we look at the citric acid cycle, this is going to take place in eukaryotic cells in the mitomatrix. So if you recall kind of the structure of the mitochondria, it has the smooth outer membrane. It has this extensively infolded inner membrane, which is also called the cristae. And the matrix is the space inside the inner membrane. So this is going to take place, this citric acid cycle, otherwise known as the Krebs cycle. I more often will call it the Krebs cycle. But again, citric acid cycle is another name. The TCA cycle is another name. There are multiple names that this process is referred to as. And so in eukaryotic cells, this is going to take place in the mitochondrial matrix. Where would you guess that this takes place in prokaryotic cells? In prokaryotic cells, still in the cytoplasm, right? They don't have a separate membrane-bound organelle to do this. So they're going to do this in their cytoplasm. So now, here we have our acetyl-CoA that was produced from the transition step. So per acetyl-CoA, when acetyl-CoA goes into the Krebs cycle, we are going to break down the sugar or break down this molecule completely. We're going to release each carbon. So notice that my product, 2 carbon dioxides. This is 1 CO2. This is a second. So we get 2 CO2s. We broke more bonds, so we're going to get more electron carriers in this step. We have 3 NAD plus going in. They're going to pick up electrons, and they're going to form 3 NADH. So 3 NADH are the products per acetyl-CoA. We also get 1 ATP by substrate level phosphorylation. And we get another electron carrier, and that is the empty one is FAD. And when it picks up its electrons and its hydrogen, it becomes FADH2. So per acetyl-CoA, we get 2 carbon dioxides, we get 3 NADH, we get 1 FADH2, which is again another electron carrier, and 1 ATP per substrate level phosphorylation. So per glucose, how many CO2s do we get? And the answer is 4, because we had 2 acetyl-CoAs. So we get 6 NADH per glucose, we get 2 FADH2s, and we get 2 ATP by substrate level phosphorylation. And so we're going to add this on to our diagram. So here is the next part of our diagram. So here we have step 2, Krebs cycle. Again, in prokaryotic cells, that's going to take place in the cytosol. So all of this still happens in the cytosol. In eukaryotic cells, this is going to take place in the mitomatrix, the innermost part of the mitochondria. And so what's going to happen again is per glucose, these are all per glucose, we have 6 NAD plus going in. They're going to pick up electrons. We get out, our product is 6 NADH. We put in 2 FADs, which is another electron carrier. What we get out are 2 FADH2s. So the FAD has become reduced. It's gained its electrons, it's gained its hydrogens, and it's energy rich. We put in 2 ADPs, adenosine diphosphates, and what we get out are 2 ATPs, and those ATPs are produced by substrate level phosphorylation. So again, it's going to be a substrate during this process that's going to transfer a phosphate group onto ADP, and that's going to form ATP. Now, the other important thing is we're breaking all of these bonds. And so notice that the other product that we get out are 4 carbon dioxide. So notice that we got 2 during the transition step and 4 during the Krebs cycle. That's a total of 6 CO2s. Does that make sense to you? And it should, because we started with a 6 carbon sugar. We should end with 6 carbons when we're done. We do. We have 6 CO2s. Those are 6 carbons. So that tells you that you did this right. You should still have 6 carbons when this is all finished. If not, you forgot some, because glucose is a 6 carbon molecule. So those are our 6 CO2s that we get. So this is going to be our Krebs cycle. Now, what I want you to really notice, though, is when you look at this diagram, look how little ATP is produced. We get 2 ATP in glycolysis. We get 2 ATP during the Krebs cycle. Yet, in the overall process of aerobic respiration, we get 30 to 32. So that means that 26 to 28 ATP are produced in the last step, meaning in the electron transport chain. So then what was the point of doing all of those earlier steps? It wasn't to yield ATP, because we didn't get much ATP. The important products that you get from the beginning of the cycle are the electron carriers. That's where the energy is. So we got 2 in glycolysis, 2 NADH in glycolysis. We got 2 NADH in the transition. We got 6 more in the Krebs cycle. And we got 2 FADH2s. So those electron carriers are actually the important products that happen during those earlier steps. So the whole idea of this is that we're breaking down the sugar little by little. And when we do this in a stepwise manner, what's actually happening is that we're transferring electrons onto these electron carriers. The electron carriers can then go drop off electrons at the electron transport chain. And when they do that, that is where the bulk of the ATP is going to come from. So again, the important products during the first 2 steps are the electron carriers. That's where the energy is. When they have their electrons, they are energy rich. And when they drop off those electrons can be used to make ATP by oxidative phosphorylation, which we're going to see in a minute. So again, what I want you to take out of this is, what is the point of this? The point of these early steps is not ATP, because we get relatively little ATP, but the purpose is break down the sugar little by little and get your electron carriers. That's where all the energy is, because the electron carriers are going to go to the electron transport chain, and that's going to be where the bulk of the energy is going to come from. So I have a question for you. Blank and blank are important, not so much for the ATP produced in them, but for the yield of electrons that are used in blank. So red says glycolysis, the Krebs cycle, and the ETC. ETC stands for electron transport chain. Yellow says glycolysis, the ETC, the Krebs cycle. Green, Krebs cycle, ETC, glycolysis. Blue, Krebs cycle, ETC, photosynthesis. Purple, the Krebs cycle, the electron transport chain, and fatty acid breakdown. So you can pause your video and you're ready. When you're ready again, push play to hear the answer. And so the correct answer, red. Glycolysis and the Krebs cycle are important, not so much for the ATP produced in them. So again, they don't produce much ATP, but for the yield of electrons that are then used in the electron transport chain. So again, we're breaking down the sugar little by little and transferring electrons onto electron carriers. Those electrons will then go to the electron transport chain to generate the bulk of the energy. So we're going to move on to step 3, and we're going to talk about the electron transport chain. So if we're talking about eukaryotic cells, right? In eukaryotic cells, this is going to take place in the cristae. So eukaryotic cells is going to take place in the cristae, which remember is the inner membrane of the mitochondria. And so what happens in this process is that embedded in that membrane are these large multi-protein complexes. And they're just numbered. So this is complex 1 on the far left, and then complex 2, complex 3, and complex 4. And so they're these large multi-protein complexes. Now, in eukaryotic cells, this occurs in the cristae of the mitochondria, meaning that these electron carrier, or these protein complexes, right? These are embedded in a membrane. Do prokaryotic cells have a mitochondria? And the answer is no. Prokaryotic cells do not have membrane-bound organelles. However, do prokaryotic cells do the electron transport chain? And the answer is yes, they do. What membrane are they going to use instead? They're going to use their cell membrane, because you're going to see in a minute that you need to have a gradient of hydrogens across the membrane. So you have to have this process spanning a membrane, and we'll come back to the hydrogens in a minute. So let's talk about how the electron transport chain works. So we have our electron carriers, right? Our NADH. NADH is going to drop off electrons at the electron transport chain. And when it drops off electrons, it's going to produce NAD+. Now it's released its passengers. It's empty. The NAD+ is ready to go back and pick up more passengers, pick up more electrons, and keep the process going. Now, NADH, when it drops off electrons, it drops it off to the first electron carrier in the pathway. So the FMN is the first one. So NADH donates electrons. So do we say that NADH became oxidized or reduced? It lost electrons, so it became oxidized, and the FMN became reduced. It gained the electrons. Now, that FMN is going to pass it to the FES, the next carrier. So it's going to pass the electrons to the next carrier. So now it was reduced when it gained electrons, FMN, but when it donates it, now it becomes oxidized and the next carrier becomes reduced. And then that carrier passes it and it goes from being reduced to being oxidized, and the process continues. So what you end up with is you end up with these alternations of oxidation and reduction as the electrons are passed down the chain. And so they're going to alternate between these oxidized and reduced states. Now, why do electrons even move down the chain? What is the force that is pulling them down the chain? Well, remember in lecture when we talked about, in the chemistry part of our lecture, we talked about electronegativity. And electronegativity is the affinity of atoms for electrons, meaning how much do they want those electrons. So if something has a higher electronegativity, it means that they have a greater affinity for electrons than they have a higher affinity for the electrons. So this carrier is more electronegative than this one, which means it has a higher affinity and it's going to pull the electrons from this one. This one is more electronegative than this one. It's going to pull the electrons. This one is more electronegative. It's going to pull the electrons. And so each of these carriers gets progressively more and more and more electronegative, which is helping pull the electrons down the chain. Now, when the electrons get to the end of the chain, we are going to have the electrons transferred onto O2. Remember we said if we're talking about aerobic respiration, O2 is our final electron acceptor. It's what's going to take the electrons from the electron transport chain. When it takes the electrons, O2 is going to become reduced, right? It gained electrons and it gained hydrogens and it formed water. So the water that we get in cellular respiration comes from O2 being reduced. O2 is going to gain electrons and hydrogens, and it's going to form water. So if you ever think about why do you have to breathe? Well, the reason you have to breathe is that you need this O2 to be the final electron acceptor, right? Something has to take the electrons from the chain. So if O2 is not available, the electron gets stuck at this one. And then the electron then gets backed up at this one because it can't pass it to the next carrier. That can't pass it to this one. So what you end up with is you end up with this backlog of electrons that can't move down the chain because they have nowhere to go. There's an electron stuck here. So you can imagine this being like a pipe, and there's a clog in the pipe. There's something that's blocking that, let's say, hairball from moving through the pipe, because if it's blocked, nothing else is going to pass. There's nothing to get it out of the way. So we have to breathe. The reason we need to breathe is we need O2 to be that final electron acceptor. And so O2 is going to be the final electron acceptor. And if it's not, right, then these electrons can't get passed down the chain and therefore can't produce ATP. So one of the things that you're going to see in a minute, electrons drop in energy as they go down the chain. And what that means is that as they're moving down our chain, they're going to give off a little bit of energy. They're going to lose a little bit of energy along the way. You're going to see in a minute that the energy that is released from the electrons moving down the chain is going to be used to pump hydrogens against the gradient. So we're going to see that in just a second. Because again, notice this part we're looking at here shows nothing about ATP production. Notice there's nothing that has to do with ATP production in this slide yet. So what we're looking at here is we're looking at the electron transport chain and how ATP is produced. So again, this is the membrane. This membrane is going to be the cristae of the mitochondria, the inner membrane. This space down here is going to be the matrix. That's where the NADH is going to be produced from the Krebs cycle. So this is the matrix. Out here in eukaryotic cells is the intermembrane space. Now in prokaryotic cells, this is a little bit different. This membrane is not the cristae, right? Because again, they don't have mitochondria. This is their cell membrane. Prokaryotic cells, this is their cytoplasm or cytosol. And this is the periplasmic space, so the space between the cell membrane and the cell wall in prokaryotic cells. So to understand the electron transport chain, one of the things that we need to put on here are the hydrogen ions. So on this side of the membrane, which let's say that we're talking about eukaryotic cells for a minute, this would be the intermembrane space. And in here would be the matrix. So notice that, again, I'm going to use this in terms of eukaryotic cells. So notice that out here, the intermembrane space has a higher hydrogen ion concentration relative to the matrix. So when my NADH comes in and it drops off electrons at the electron transport chain, again, NADH becomes oxidized. It's going to produce the NAD+. NAD+ can go back and it's going to pick up more electrons to bring back to the electron transport chain. So NADH comes along. It's going to drop off electrons at the electron transport chain. Now, this carrier is more electronegative, the FMN, and then it's going to pass it to the next carrier. And remember that each of these carriers becomes more and more and more electronegative. So as the electrons go down the chain, remember that they're going to give up a little bit of energy. They're going to lose some energy along the way. And what happens is that the energy that it's giving off is used to pump hydrogens against the gradient, meaning it's going to pump hydrogens from a low concentration to where it's high. Do molecules normally want to move from low to high spontaneously? And the answer is no, right? If I have a beaker of water and I add food coloring to that beaker, the dye is going to go from the high concentration, meaning the drop, to the low concentration, which is the water around it. It's never going to be all spread out and spontaneously go back to being a drop. Not going to happen. Going from low concentration to high is not a spontaneous process. So hydrogens don't want to move from low to high. Something has to drive that movement. And what is powering the hydrogens to be pumped across the gradient, or against the gradient, I should say, is that those the electrons, as they go down the chain, they give off energy. And it's that energy that's used to pump the hydrogens against the gradient. So the energy to pump the hydrogens comes from the electrons. Now, again, O2 is going to be our final electron acceptor, right? It's what's going to take the electrons from the chain, and it's going to take the electrons and it's going to reduce the O2 to form water. Now again, that part is still not showing us anything about ATP production. So where does the ATP come from? Well, if you look at this blue molecule over here, this is ATP synthase. The name tells you what it does. ATP synthase is used to synthesize ATP. It's going to make ATP. How does this work? Well, hydrogens are now going to flow back down the concentration gradient. Now hydrogens are going to go from high to low. And when they go from high to low, that movement is favorable, right? Things want to go from high to low. That is a spontaneous process. That is energetically favorable. So what happens is when the hydrogens go back down the gradient, that movement of hydrogens through ATP synthase causes this rotor to spin. It causes this rotation. And that rotation, that movement, when it causes the rotor to spin, causes these catalytic knobs, this bottom part, to move. We have now mechanical motion, right? And so these catalytic knobs are going to move, and that kinetic energy, that energy of motion, is going to be used to produce ATP. So this type of ATP production, again, is referred to as oxidative phosphorylation, because we're getting ATP not from transferring a substrate or a phosphate from a substrate. In this case, we're producing ATP because of these redox reactions. So the electron transport chain, which is basically a big redox reaction when we donate electrons from one carrier to the next, when they get passed along the chain, that pumps hydrogens against the gradient. So it's going to pump the hydrogens from low to high. Those high concentration, now we have an even greater hydrogen ion gradient, meaning we have more hydrogens in the space out here. Hydrogens are going to go back down. They're going to go from high to low. That is energetically favorable. That is going to produce the ATP. And so this is where the bulk of the ATP comes from. Now back to why oxygen has to be your final electron acceptor. Because what happens is if O2 is not present, the electron gets stuck here. It has nowhere to go. The electron gets stuck here. It gets stuck here, all along the chain. The electrons can't move from the carrier they're at. If they can't move, we can't pump hydrogens, because the electrons moving down the chain is what's giving off energy, and that energy is what's pumping hydrogens against the gradient. So notice that without O2, we can't pump hydrogens against the gradient. If we no longer have that gradient, if we no longer have that high concentration of hydrogens, the hydrogens are not going to flow from high to low because that gradient is disrupted, and the cell's not going to be able to produce ATP. And this is why if you go without oxygen for too long, you end up dying, because your cells need that O2 in order to make ATP. Without it, they're not going to make enough ATP for you to survive. Now, let me give you a little bit of an analogy about how ATP synthase works. I do have some videos that I'm going to include on Canvas that are good study tools for studying cellular respiration. And it kind of helps illustrate how this ATP synthase, how it rotates. And so it's good to watch and kind of see how ATP synthase works. But let me kind of describe it for you. So if you've ever seen a dam, right? So here's our dam and here's our water. And so the water is behind the dam, and then there is a penstock, this tubing or this opening, where when they open the dam, the water can go into this penstock. Now at the bottom is a little turbine that spins. So if the dam is opened and the water goes down, notice it's going from higher up to lower. It's going from high to low. And that movement of water causes that turbine at the bottom to spin. So the flow of water is going to cause that turbine to spin. And when that turbine spins, that generates electricity. So that's how electricity can be generated, right? We're using water, the movement of water, to cause the turbine to spin. If you've ever seen the windmills on the way out to Palm Springs, those are using wind energy. They're using wind to cause those turbines to spin, and that is used to generate electricity. Same idea here. ATP synthase, think of like a turbine. Instead of water flowing across it, though, it's the movement of hydrogens. So it's the hydrogens going from high to low that's causing this turbine to spin. And when this turbine is spinning, notice it has this arrow like it's going in a circle. When that turbine is spinning, when that rotor is moving, that creates energy because that is kinetic energy. It's energy of motion that causes these catalytic knobs to move, that energy is now going to be transferred to take ADP and produce ATP. So it's that energy of motion that is going to produce the ATP. And that motion comes from redox reactions, right? Because the redox reactions are what are driving the hydrogen ion gradient. So the energy stored in the hydrogen ion gradient across the membrane couples the redox reactions of the electron transport chain to ATP synthesis, right? And so these two types of reactions are coupled. And the hydrogen ion gradient is referred to as proton motive force, meaning we use protons, motive referring to motion, proton motive force, meaning that the hydrogen ions, that gradient and then moving from high to low is basically going to do work because the motion of those hydrogens going from high to low is going to cause ATP synthase to move. And so again, we call this process oxidative phosphorylation. We're using redox reactions to generate that hydrogen ion gradient, and then we're going to use that hydrogen ion gradient to produce ATP. So this is just looking at more detail of ATP synthase. So this is our ATP synthase that you're looking at here. Notice the top part has a rotor. So here's the rotor, and the rotor can rotate. And so the intermembrane space has the higher hydrogen ion concentration. And that's because the electron transport chain, you can think of it being over here, the electron transport chain is going to pump hydrogens into the intermembrane space. So we have a higher hydrogen ion concentration here and a lower concentration in the mitochondrial matrix. Let's put all this on here. So as the hydrogens move back across the membrane, meaning when they go back from their high concentration to low, it's going to pass through these channels in ATP synthase. And it's going to cause this rotor to move. So the hydrogens are going to go down the gradient. It causes this rotation of the rotor, which then is going to cause these catalytic knobs to move. They're going to move out and in. And that is going to catalyze the production of ATP. So what happens is ATP synthase is going to use the exergonic flow of hydrogens. Exergonic meaning energy is released because going from high to low is energetically favorable, right? Things want to go from high to low. So that is exergonic. It's a favorable reaction. It's going to release energy. When that energy is released, that energy is transferred onto forming ATP, right? Going from ADP in an inorganic phosphate to form ATP, that step is endergonic, right? It requires the input of energy to form that bond. Remember that phosphate groups do not want to be together. So to add this phosphate onto adenosine diphosphate is not easy. It's not favorable. It's not something that's going to happen spontaneously because it takes a lot of energy to link those phosphates together. So again, this shows us a coupled reaction. We have an exergonic reaction, the hydrogen's flowing down the gradient, coupled to the endergonic reaction of forming ATP. So this is just another example of a coupled reaction. You have to get energy from somewhere. And in this case, it's hydrogens going down the gradient that is providing the energy to make the ATP. And so this is an example of what we call chemiosmosis. If you know what osmosis is, it's the movement of water. In this case, it's chemiosmosis. So we're using chemicals, hydrogen ions. We're using the energy and hydrogen ion gradients to drive cellular work. We're using that gradient to produce our ATP. So let's continue on with our diagram. So step number 3, we have on here our electron transport chain, right? Here's my electron transport chain. In prokaryotic cells, remember that this is going to be the cell membrane. In eukaryotic cells, this is the cristae, which again is the mitochondrial inner membrane. I went ahead and I labeled both of these spaces depending on if it's prokaryotic or eukaryotic. Prokaryotic, I kept pink. So this would represent the space out here is the periplasmic space in prokaryotic, or in eukaryotic cells, it's the intermembrane space, the space between the inner and the outer membrane. This space in here, that is the cytosol or the cytoplasm in prokaryotic cells. But in eukaryotic cells, it's the mitochondrial matrix, meaning the innermost part of the mitochondria. So what is going to happen in the electron transport chain? So again, I'm going to draw this on here. NADH is going to drop off electrons at the electron transport chain. And so again, when it drops it off, NADH becomes oxidized, it loses its electron, and we're going to get NAD+. So we have our NAD+, but the electrons are going to move down the chain. FADH2, remember that was the other electron carrier that was generated during the Krebs cycle. FADH2 will also come in and drop off electrons at the electron transport chain. And when it drops off electrons, it becomes FAD. So electrons go down the chain. What is going to take the electrons from the electron transport chain if this is aerobic respiration? What is going to remove the electrons? Answer, oxygen. O2 is our final electron acceptor. It's going to take the electrons, it's going to become reduced, and it forms water. You don't need to worry about balancing that equation. I don't care. I just want you to know that O2 is the final electron acceptor. And when it does that, it's going to form water. So there's our water. Now, as the electrons go down the chain, they're used to pump hydrogens against the gradient. So we're going to pump hydrogens from the low concentration to the high concentration. So we're going to pump these hydrogens. And so the hydrogens will go against the gradient. We're going to make an even higher hydrogen ion concentration in the intermembrane space or the periplasmic space, depending on which cell type you're talking about. Then hydrogens go back down the gradient. They go from high to low. And that process across ATP synthase, that is energetically favorable, right? Going from high to low is favorable. And it's the flow of the hydrogen ions down the gradient that causes ATP synthase to move, which is going to generate ATP by oxidative phosphorylation. In terms of the number of ATP, remember that from this step, we get about 26 to 28 ATP. So that is where the bulk of the energy is going to come from. The bulk of the energy is going to come from oxidative phosphorylation. And so again, this is where you can see that those early steps are important for those electron carriers, because you need the electron carriers to drop off the electrons. You need the electrons to move across or down the electron transport chain, because it's the movement of the electrons that pumps hydrogens against the gradient. You need that hydrogen ion gradient in order to produce the ATP. So again, those early steps, big picture here, again, I want you to see the big picture. The early steps are important because they generate the electron carriers. Those electron carriers are then used in the electron transport chain. And that is where the bulk of the ATP is going to come from. So again, when you study this, you want to study for each step what goes in, what comes out. And I would recommend, just to practice studying, just keep drawing this out. Keep trying to draw this out by memory. If you can draw this out by memory, you can answer any question that I have related to this, because that means that you understand the process. And that's what I want you to take away from this, is I want you to walk away with an understanding of why does it work this way? What is the point of all this, right? And the point of all of this is to break down the food little by little. And when we do that, we release electrons and they get put on the electron carriers. Electron carriers go to the electron transport chain. That's going to produce the bulk of the ATP. So that's the point of this, right? Now, when we talk about aerobic respiration, notice that we have glucose. Is glucose or sugar the only food source that we use? Answer is no, right? Glucose is one food source, right? We can use lipids, we can use fats, we could use proteins. Those other types of macromolecules can get converted into different components that can come in at different points in this pathway. So what you need to realize is that while we draw this with aerobic respiration using glucose, glucose is not the only food source. Other types of food that we eat can also help drive this process. It just does it in a slightly different manner. So let's summarize what goes in and what comes out during the electron transport chain. So what goes in? NADH. Right? It's going to drop off electrons at the electron transport chain. What is going to come out? NAD+. We have our other electron carrier, FADH2, comes in. FAD is what comes out. It's not plus in this case, it's just FAD is what comes out, because again, it's when the electrons get dropped off at the electron transport chain. What comes out, those empty electron carriers are free to go back and pick up more electrons. What goes in, we need O2. What comes out from the O2? Water. So when the oxygen acts as the final electron acceptor, it's going to have water being produced. And then lastly, we have adenosine diphosphate plus inorganic phosphate. And what comes out? ATP. Whoops. Adenosine triphosphate. That's our product. And so again, this is just summarizing for the electron transport chain, what goes in and what comes out. So this slide is summarizing cellular respiration. And so again, the first step is going to be glycolysis. Glyco refers to sugar. Lysis is breaking. So we take that 6-carbon sugar and we chop it in half to 2 3-carbon pyruvates. Now, when we break these bonds, we transfer electrons onto electron carriers. So during this process, we also get 2 NADH. So here are our 2 NADH. we get 2 ATP, and the ATP produced during glycolysis is by substrate level phosphorylation. We simply take a substrate that has a phosphate and we transfer it on to ADP, forming ATP. Those 2 pyruvates or pyruvic acids will go into the mitochondria and they will go through a transition step. And so during that transition step, for those 3 carbon pyruvates, they're going to break off 1 carbon. So you end up at this transition step with 2 CO2s. And to the remaining 2 carbon fragment, you're going to add a CoA on. And your product is going to be acetyl-CoA. 2 of them. Now, again, we're breaking some bonds, so we get some electron carriers. So during this process, we also get 2 NADH. Acetyl-CoA is going to go into the Krebs cycle, which is also known as the citric acid cycle. And during this process, we're going to break down the acetyl-CoA completely. And what we get out is we get 4 carbon dioxides. We also get a little bit of ATP, so we get about 2 ATP per glucose, and this ATP is produced by substrate level phosphorylation, just like in glycolysis. We broke a lot more bonds, so we get a lot more electron carriers. We get 6 NADH, we get 2 FADH2s, and then these electron carriers are now going to go to the electron transport chain. And so again, just to remind you, these early steps are not so critical for the ATP because we get very little ATP during these early steps. We only get 2 in glycolysis. We only get 2 in the Krebs cycle. The important product of the earlier part of this pathway is actually going to be these electron carriers because those electrons are energy rich. And when they go and drop off electrons at the electron transport chain, so when they drop off these electrons, the electrons are going to go down the chain, and as they go down the chain, that's going to be used to pump hydrogens against the gradient, pumping hydrogens from a low concentration to high. Hydrogens will go back down the gradient through ATP synthase, and that movement of hydrogens across the membrane is what's going to power the production of ATP. And so during the electron transport chain, we get about 28 ATP through oxidative phosphorylation. So using those redox reactions to make ATP. And so we get about 28 ATP during this process. So total, we get about 32 ATP. Now, you might wonder, sometimes you'll see it written, we get 30 to 32. And you might wonder, well, what dictates if you get 30 or 32? And it has to do with in eukaryotic cells. So notice that in glycolysis, we make these NADH out in the cytoplasm. The NADH needs to drop off electrons to the electron transport chain, which happens inside the mitochondria. And so what actually happens is that the electrons have to get shuttled across the membrane. So the NADH itself actually does not come in. Instead, it's going to have a shuttle here, and those shuttles are going to transfer electrons to empty electron carriers in the mitochondria. Now, different cells in the body have different types of electron transporters. In some cells, the electrons get transferred onto NAD+, and we get NADH. In other cells, there is an FAD transporter, meaning that the electrons from the NADH actually get put onto FAD and form more FADH2. Now, why does that matter? NADH comes in at complex 1, FADH comes in at complex 2. So when we use NADH, we get more ATP than if we use FADH2. I won't ask you that, but just in case you're wondering where that difference comes from. So now we're going to fill in and just review where these different steps take place. And so if we're talking about glycolysis in eukaryotic cells, where does that take place in eukaryotic cells? And the answer is the cytosol. In prokaryotic cells, it's also the cytosol. The intermediate or transition step in eukaryotic cells is going into mitochondria. In prokaryotic cells, cytosol. The Krebs cycle. The Krebs cycle in eukaryotic cells is going to take place in the matrix, so the innermost part of the mitochondria. In prokaryotic cells, it still happens in the cytosol. If we get to our electron transport chain, in eukaryotic cells, this happens in the mitochondrial cristae. So again, that's the inner membrane of the mitochondria. Prokaryotic cells, however, don't have mitochondria, so they can't use that membrane. What membrane do prokaryotic cells use to do their electron transport chain? And the answer is they use their cell membrane. And so back when we talked about different structures within the cell, you might recall that when we talked about the cell membrane, we said that in prokaryotic cells, one of the functions of the cell membrane is that it's used to produce ATP. And so that is true in prokaryotic cells. In prokaryotic cells, the cell membrane is used to make ATP. In eukaryotic cells, this electron transport chain is going to happen in the cristae of the mitochondria. So, question for you. Breathing is necessary because we need red, both atmospheric nitrogen and the oxygen for energy transformation. Yellow, oxygen to donate electrons to the electron transport chain. Green, nitrogen to donate atoms to oxygen. Blue, oxygen to act as the final acceptor of electrons in the electron transport chain. Or purple, oxygen to donate phosphate groups to ADP, making it ATP. And so when you're ready, go ahead and pause the video and think about your answer. And then when you're ready, go ahead and push play to hear the correct answer. So if you said blue, you are correct. Breathing is necessary because we need oxygen to act as the final electron acceptor in the electron transport chain. That's what's going to take the electrons from the chain. And that allows the electron transport chain to occur because we need the electrons to be able to move in order to pump hydrogens against the gradient and then to make ATP. So we need to breathe because we need O2, oxygen, as our final electron acceptor. It's not red, both atmospheric nitrogen and the oxygen for energy transformation. That is not true because most organisms actually cannot use atmospheric nitrogen, which is N2 gas. There are a few exceptions. Some organisms can be nitrogen, can do a process called nitrogen fixation. But again, most organisms don't utilize atmospheric nitrogen, so that one's not true. Oxygen to donate electrons to the electron transport chain, that's not true. They're not going to donate, they're going to receive. What molecule is going to donate electrons to the electron transport chain? And the answer is it's the NADH and the FADH2. Those are what are going to drop off electrons to the electron transport chain. Where do those electrons come from initially? They come from the food that we eat, meaning they come from when glucose becomes oxidized as we break down the glucose. But directly, they come from NADH and FADH2. Green, nitrogen to donate atoms to oxygen. That's not true. That one doesn't even make sense. Purple, oxygen to donate phosphate groups to ADP, making it ATP. O2 does not have phosphate groups, therefore it can't donate them to ADP. So on Canvas, I've posted some videos for you to watch. One video comes from the textbook and it's an overview of metabolism and it walks through the different steps. And the second video is called Oxidate It or Love It. And it's basically students from Stanford who made a rap talking about cellular respiration. And it might be a helpful way to help you learn about cellular respiration, because oftentimes it's easier to remember a song than it is to just study something on a paper. I know for myself, when I hear the actual song, like on my phone, I think of the lyrics from this video. So it does actually kind of stick in your head, and it might help you remember cellular respiration a little bit more. And so that video is also on Canvas, and I put the link here as well. So we talked about aerobic respiration. Now we need to talk about anaerobic respiration. And again, anaerobic respiration does not use O2 as the final electron acceptor. Does use inorganic final electron acceptors, right? So they do use inorganic final electron acceptors like nitrate, sulfate, carbonate, etc. And so we saw this in lab when we looked at our nitrate reduction test, right? Nitrate can be reduced to form nitrite. Now, in that test, when we're doing that test, essentially what we're doing is we're trying to determine if bacteria can do anaerobic respiration. Can they use a final electron acceptor that is not O2, but is instead nitrate? And so that test was testing that. In terms of sulfate, sulfate gets reduced to form hydrogen sulfide. What test did we do that looked at the production of hydrogen sulfide? And the answer is we did our TSI test, right? Our TSI test was what we used to determine if sulfur was reduced, meaning did sulfate get used as the final electron acceptor? So when we talk about anaerobic respiration, we still have glycolysis. We still have the same intermediate and transition step, but we have a slight variation in terms of the Krebs cycle and the electron transport chain. So while the majority of the process is the same, they're not identical. And so as a result, gives about 3 to 29 ATP per glucose. So during anaerobic respiration, we get more ATP than fermentation, but we get less than aerobic respiration. So in terms of the types of organisms that do anaerobic respiration, we have Pseudomonas, and Bacillus can use nitrate. And you might recall back to when we did our nitrate reduction test, Pseudomonas even took it a step further. It did not just produce the nitrite, but it fully reduced it and produced gas in the Durham tube, which then the nitrogen gases were produced. So the atmospheric nitrogen, so either N2, nitric oxide, nitrous oxide, etc., could be produced. And so those are some examples of organisms that can do anaerobic respiration. So this slide is comparing and contrasting respiration and fermentation. And so in both respiration and fermentation, we still go through glycolysis. So notice that's right down the middle. That part is the same. So in glycolysis, we are going to chop glucose in half to 2 3-carbon pyruvates. And when we do that, NAD plus is going to pick up electrons and it's going to form NADH. So during glycolysis, we form NADH. We also get a little bit of ATP. We get 2 ATP per glucose. Pyruvate serves as this key juncture between whether the cell is going to do respiration or fermentation. If O2 is present, we're going to go through the rest of respiration. If there's no O2, the cell can switch over to fermentation. Now, if we look at this process, right? So notice if we look at respiration, if we use respiration, we use inorganic final electron acceptors, right? So O2, if we're talking about anaerobic, it would be nitrate, sulfate, carbonate, etc. Those are all inorganic final electron acceptors and produces inorganic products, meaning it's going to break the glucose down completely to CO2. So during respiration, we're going to have our Krebs cycle, we have the electron transport chain, we use our inorganic final electron acceptor, and we're going to produce inorganic products, meaning we break it down completely. The sugar is broken down completely to carbon dioxide. Now, in order for respiration to continue, notice that glycolysis in the beginning, we need NAD plus to be able to take electrons during the process of glycolysis, right? NAD plus is going to pick up electrons and it's going to become NADH. And so I said that all of these early steps are important, not for the ATP that's produced in them, because we get actually very little ATP, but the important thing that's produced during the early steps are those electron carriers. And so those electron carriers will go to the electron transport chain. They will drop off electrons. And when they drop off electrons, that's going to produce the bulk of the ATP. Now, in order for this process to keep occurring, when those electron carriers drop off their electrons, they are going to go back, right? So they will form the NAD+, or they will form the FAD. So when they drop off electrons, it's back to being NAD+. Now the NAD+ can go back and pick up more electrons. So in respiration, the way that we regenerate the NAD+ to keep this going is that the electron carriers need to be able to drop off electrons at the electron transport chain. So again, if O2 is not available, right? If O2 is not available, then the electron transport chain can't go. And if the electron transport chain can't go, we can't get ATP, and we get this buildup of these reduced electron carriers, meaning we get a lot of NADH and FADH and FADH2 in the cell because they can't drop off their electrons. We would run out of NAD+. So if we compare that to fermentation, fermentation uses organic final electron acceptors and produces organic products. So fermentation is a little bit different. It uses organic final electron acceptors, either pyruvate or acetaldehyde, depending on if it's lactic acid fermentation or alcoholic fermentation. But we will use organic final electron acceptors, and as a result, we also produce organic products, ethanol or lactic acid. They still have a lot of energy left. Now, during the process of fermentation itself, so meaning going from pyruvate to form lactic acid, that part of the pathway actually doesn't generate ATP. So when we talk about fermentation, the ATP that gets produced actually comes from glycolysis, because no matter which type of sugar metabolism that they do, glycolysis is going to occur. And so in fermentation, they get their ATP from glycolysis. So if that's true, why do we even have to have the fermentation part? Why do we have to use pyruvate, for example, as the final electron acceptor? And the answer is that in order for this to occur, notice that in respiration, the NAD plus gets regenerated when the NADH drops off electrons at the electron transport chain. That's how we get back to having NAD plus, so that this process can keep continuing. In fermentation, we don't have an electron transport chain, so we can't use that method to regenerate the NAD plus. So in terms of fermentation, the purpose of fermentation is to generate ATP and, the key here, and regenerates NAD plus. NAD plus needs to be produced so that glycolysis can keep happening. So NADH is going to drop off electrons to pyruvate, and when it does that, it forms the NAD plus. And then now the NAD plus can go back and can be used in glycolysis so that at least glycolysis can continue, so that we can get the ATP during glycolysis. So when we talk about fermentation, again, you want to see the big picture. What is the point of doing this? And the point is that if O2 is not available and the organism is not doing an electron transport chain, they still need to be able to do glycolysis to get their ATP. And so in order to keep glycolysis going, we need a way to regenerate NAD+. And so again, the way that we do that is that we use organic final electron acceptors like pyruvate, like acetaldehyde, and we're going to use those organic products to take the electrons so that NADH can go back to being NAD+, and NAD+ can go back to glycolysis and pick up more electrons, so that at least glycolysis can occur and at least we can get some ATP during this process. It's not much, it's only 2 ATP per glucose, but it works well enough for slow growing organisms. So now we're going to move on and we're going to talk about fermentation. So when we talk about fermentation, fermentation can be a very broad term used to describe a variety of processes. It can be used to describe any spoilage of food by microorganisms. So that would be a general use. It's any process that produces alcoholic beverages or acidic dairy products. That would be a general use. So a yeast, for example, Saccharomyces cerevisiae. Saccharomyces cerevisiae translates to sugar fungus that makes beer. That's what their name means, sugar fungus that makes beer. And so when Saccharomyces cerevisiae is grown under anaerobic conditions, meaning it's grown in this chamber where no O2 can get in, that organism is going to switch to fermentation. And specifically, it's going to switch to alcoholic fermentation. And so this is a way that alcohol can be produced. Fermentation can also be used to describe any large-scale microbial process occurring with or without air. So that's a common definition used in industry. You might also recall that your lab book uses the term fermentation very loosely. So for example, when it's talking about a phenyl red broth test, when it's talking about a phenyl red broth test, the purpose, it says, is to determine if bacteria can ferment a specific sugar. However, I was careful to change it to can bacteria metabolize a specific sugar? And the reason is that your book, when it talked about your lab book, when it talked about fermentation, it was just talking about the metabolism of that sugar. It wasn't necessarily referring to fermentation specifically. Think of our phenyl red broth. That environment was not anaerobic, right? It was a liquid broth. It was in the incubator. So that process wasn't necessarily true fermentation. So fermentation, you have to kind of watch how it's used. But in the context of this class and what we're learning in terms of metabolism, when I talk about fermentation, I'm referring to the anaerobic process of fermentation. And you're going to see what that looks like in just a minute. So fermentation is an anaerobic process, but is not anaerobic respiration. And so again, there's a key distinction. You'll see this online, you'll see this in some books where they call fermentation anaerobic respiration. That is incorrect. And the reason is that in fermentation is using a final electron acceptor that is organic and produces organic products. That's different than an anaerobic respiration. Anaerobic respiration breaks down the glucose completely to inorganic products like carbon dioxide. And in anaerobic respiration, we have an electron transport chain. And we also have, in the case of anaerobic respiration, the final electron acceptor is inorganic. But again, in fermentation, the final electron acceptor is organic. So again, make sure that you don't get tripped up on fermentation and anaerobic respiration. They're totally different processes. In the case of fermentation, we get 2 ATP, and those 2 ATPs come from glycolysis. And so the ATP produced during glycolysis comes from substrate level phosphorylation. So again, it's simply going to be that during glycolysis, a substrate has a phosphate and it's going to transfer it onto ADP, forming ATP. So fermentation is going to use our substrate level phosphorylation. The fermentation itself starts with the end products of glycolysis, meaning that it starts with pyruvate. So it starts with the pyruvate. And again, during fermentation, we get incomplete oxidation of glucose, meaning that the glucose is not broken down fully, and we end up with organic products. They still have a lot of energy left. So pyruvate serves as the key juncture between whether the cell is going to do fermentation or whether it's going to do cellular respiration. So if O2 is present, pyruvate will get transferred into the mitochondria. It will go through the transition step. It'll go through the citric acid cycle step. It will go through an electron transport chain. And again, the purpose is, in this case, right? So to do acetyl-CoA and the Krebs cycle, you need to have O2, because in order to do the electron transport chain, you need O2 to be the final electron acceptor. So if O2 is available, pyruvate will go into the mitochondria. However, if O2 is not available, right, if O2 is not present, then the cell's not going to transport the pyruvate into the mitochondria, because without O2, the electron transport chain can't occur. So in that case, if there's no O2 present, the organism is going to switch to fermentation. And fermentation is going to be an anaerobic process. Again, it does not mean it's anaerobic respiration, but it will switch to fermentation, which is an anaerobic process. And so what's going to happen is that in fermentation, the pyruvate itself can act as the final electron acceptor if we're talking about lactate as the product, or pyruvate can be converted to acetaldehyde. Acetaldehyde acts as the final electron acceptor, and it produces ethanol. So pyruvate is kind of this key decision between whether the cell will do aerobic cellular respiration or whether it will do fermentation. So we're going to look at the types of fermentation. And the first type of fermentation that we're going to talk about is our alcoholic fermentation. And alcoholic fermentation occurs in yeast cells like Saccharomyces cerevisiae. It's also used by some bacteria. And so the way that this works is that we still have glycolysis. So notice here's my glycolysis. Glucose is going to get broken down into that 3-carbon pyruvate, right? Glyco, sugar, lysis breaking. When we break down the glucose, electrons are going to be transferred onto an electron carrier. So NAD+ is going to take electrons and it's going to form NADH during glycolysis. We also get a net gain of ATP of 2 ATP per glucose during glycolysis. And again, the purpose of doing fermentation, so purpose, regenerate, to regenerate NAD+. And so what ends up happening is that pyruvate is going to break off a carbon and we get carbon dioxide. This remaining 2-carbon molecule is called acetaldehyde. Acetaldehyde is going to be our final electron acceptor. It's what's going to take the electrons from the NADH. So during the fermentation part, acetaldehyde is going to accept the electrons, right? So this NADH is going to donate electrons to the acetaldehyde. And when it donates its electrons, it goes back to being NAD+, right? Because it's losing its passengers. So it's back to being NAD+, and then that NAD+ can go back and do glycolysis so that at least the cell can make 2 ATPs per glucose. The product during this process is going to be alcohol, ethanol. So this is our product. Notice that my product is organic because it has carbon and hydrogen. My final electron acceptor was organic because it had carbon and hydrogen. So we end up with an organic product and we have an organic final electron acceptor. And so this is what fermentation looks like. There's no electron transport chain. That's why it's not called respiration. And the purpose is that we need to regenerate the NAD+. That's the main purpose because we need to keep the NAD+ coming so that at least the cell can continue to do glycolysis and make ATP. Because you can imagine that if you don't regenerate NAD+, right? Normally, during respiration, the NADH drops off electrons at the electron transport chain. And that's what allows the NAD+ to be regenerated. If, however, we don't have an electron transport chain because there is no oxygen, right? If we were just doing glycolysis to get ATP and that was it, that's a problem, because as the cell continues to do glycolysis, it's going to keep producing NADH, but not having any more empty NAD+, because all the NAD+ is being utilized to form the NADH during glycolysis. So in fermentation, we need to regenerate the NAD+. That's pretty much the goal of fermentation, because without oxygen, we're not using an electron transport chain. So cells have to have an alternate way to regenerate the NAD+ so that at least glycolysis can continue to occur. So the next type of fermentation that we're going to talk about is going to be our lactic acid fermentation. And lactic acid fermentation occurs in animal cells, in the muscles, for example, and in some bacteria like lactobacillus, which is used to make yogurt. So these organisms have a different set of enzymes to do a different type of metabolism. And so they're able to do lactic acid fermentation. So again, we still have glycolysis. And glycolysis, again, we're going to break down glucose into the 2,3-carbon pyruvates. That process is also going to transfer electrons onto NAD+, and we get NADH. We also get 2 ATP by this process. Again, goal of fermentation, regenerate the NAD+. And so in this process, pyruvate acts as the final electron acceptor. Again, it's organic. And so pyruvate itself is going to be our final electron acceptor. And so when it takes the electrons, right, and it becomes reduced, it's going to form lactate, otherwise known as lactic acid. Now, lactic acid you might hear about for a specific reason. If you think of like being an athlete and working out or playing sports. Now, lactic acid is thought to be, although there is some debate, lactic acid is thought to be responsible for the reason why your muscles burn after a hard workout. And that's because if you think about it, right, if I'm exercising really hard, if I'm doing a lot of cardio and I'm exercising really hard and I'm breathing heavy, if I'm breathing heavy, I might not be getting enough oxygen to my tissues. And so muscle cells need ATP in order for the muscle cells to contract, because they need to hydrolyze ATP for muscle contraction. So if those muscle cells are not getting any oxygen, then those muscle cells are going to switch to fermentation, at least in the short term. And when they switch to fermentation, they produce lactic acid. Now, that lactic acid could build up in the muscle cells, and that might be why muscles burn after a hard workout. Now, if you think about it, does that last forever? Do your muscles permanently burn like that? No, because that lactic acid is still energy rich. It still has some energy in it, and it can be put back into metabolism and broken down further. And then that muscle burn will subside. And so again, the purpose of fermentation is to regenerate the NAD+ so that at least glycolysis can continue and we can at least get 2 ATP during this process. So in lactic acid fermentation, pyruvate is reduced by NADH to form that lactic acid. Sometimes other acids may result. If we call the organism homolactic, homo means same, and that means that only lactic acid, make that L better, oops, only lactic acid is produced. So example, lactobacillus is used to make yogurt. Other organisms might be what we call heterolactic. Hetero means other. So for these organisms, they produce lactic acid plus other acids. So they have a slight variation, and they don't only produce lactic acid, but they might produce other acids as well. Swiss cheese, for example, would contain propionic acid. Sourdough is sour because there are bacteria that produce acids that make the bread sour. And so we have heterolactic. Heterolactic means that they form lactic acid and other acids as well. And in some cases, gas may also be produced. Now, if you go back and you look at the slide for alcoholic fermentation, notice that when we talked about alcoholic fermentation, one of the products was CO2. CO2 is what's responsible for making beer carbonated, for example, right? And so that CO2 is going to carbonate the beverage. And so that's why when you pour beer, you might get some foam. It's the carbon dioxide that was produced by the yeast when they did the alcoholic fermentation. The other thing that I wanted to point out about that gas is that the yeast is also used to make bread. And the reason that the yeast is used to make bread is because if the yeast is anaerobic and they do that alcoholic fermentation, they're going to produce CO2. That CO2 is going to help the dough to rise. And so that's why yeast is also used to make bread. Now, if I take that dough that has risen and I bake it, am I going to be intoxicated by alcohol that might be in the bread? And the answer is no, right? Because when you bake that dough, alcohol evaporates pretty quickly. And so the alcohol will evaporate off. And so that's why you don't get drunk, for example, or intoxicated when you eat bread. There have been instances where dogs, for example, have gotten into dough that somebody's made. And if a dog eats that dough before it's been cooked, yes, the dog can become intoxicated because the alcohol is still in the dough. But when you bake it, it's not a problem because the alcohol will evaporate off. So this is just a slide summarizing respiration and fermentation. And so what I'm going to do is I'm going to explain to you what I'm looking for in this table. And then I'm going to actually have you do this as a participation assignment. So I will go into Canvas and I will change one of the participation assignments to be a submission. You can either print this slide and fill it in, or you can draw your own table on a separate sheet of paper. And then you're just going to upload your filled in table into Canvas, so into that assignment. Just like you guys are doing for your parasite video, for example, right? You're filling out the questions and then you're uploading them into Canvas. Same idea for this. This is going to count, though, as a class participation. So when the due date closes for this, then I will post the answers so that you're studying the right material. So what we're looking for in this table is we are looking at how many ATP are produced. So in glycolysis, so we'll walk through this together for the first row. In glycolysis, how many ATP do we get? And the answer is 2. We get 2 ATP. Is O2, is oxygen required for glycolysis to happen? No. And that's because glycolysis happens whether we're talking about aerobic respiration, anaerobic respiration, and fermentation. Phosphorylation is basically how ATP is made. So what is the mechanism that ATP gets made during that step? So if we're talking about glycolysis, what is the method that ATP gets produced during glycolysis? And that is, it's produced by substrate level phosphorylation. So the method that it's using is substrate level phosphorylation. In terms of final electron acceptor, there's a couple ways that you could interpret this. You could interpret this that there is no final electron acceptor in glycolysis because it's going to keep going to later steps. Or you could interpret this as what is taking the electrons during this step. So if I'm interpreting this as what is taking the electrons during this step, what is accepting those electrons, that's going to be the NAD+. Whoops. NAD+. And it's going to form NADH. So the electrons from the food are going to get transferred on to NAD+, and it's going to form our NADH. So we get our NADH from this process, and that's what is taking, so the NAD+ is what's taking the electrons during this step. So you're going to fill in this table, the rest of this table for your participation. These top three are assuming we're looking at aerobic. So you're going to fill this in for the Krebs cycle during aerobic respiration, the electron transport chain for aerobic respiration. The next row is looking at anaerobic respiration, and this is including all steps. So you're going to combine them all together. So that includes glycolysis, Krebs cycle, and electron transport chain all combined. So how many ATP does the entire anaerobic respiration produce? Is O2 required? How is the ATP produced? Hint, there might be more than one. And then what are the final electron acceptors during anaerobic respiration? The last row is going to be fermentation, right? And we're going to, in this part, include glycolysis, right? Because glycolysis is where the ATP is going to come from. So make sure that when you're talking about fermentation that you're also including glycolysis as part of your answer when answering that row.